Hi children, let us come to the another video here, and the name of the chapter which we will cover here, that is molecular basis of inheritance. और इससे पहले हमने जो चैप्टर डिस्कस किया दैट इज प्रिंसिपल्स ऑफ इनहेरिटेंस एंड वेरिएशन, जो मैंने 7 वीडियोस में कवर किया. नाउ इस चैप्टर में हम कैरेक्टर्स के ट्रांसमिशन को मॉलिक्यूलर बेसिस पे समझेंगे. How genes they control the characters, and genes they are nothing but the DNA segments that we know. Actually, DNA से RNA बनता है और RNA से फिर protein. This is the whole process. Because gene it codes for protein which is responsible for a particular character. DNA is a genetic material that we know. कब से पढ़ते आ रहे हैं that DNA it is the genetic material. But RNA is also a genetic material. ये भी हमें पता है. अब डिफरेंस ये है कि RNA it is a genetic material which is not in good number in nature. It is present in some viruses actually, which are called as retroviruses. और RNA एक stable genetic material नहीं है. एंड RNA वर्ल्ड जो कि इसी चैप्टर का एक टॉपिक है, उसमें हम ये भी पढ़ेंगे कि DNA इट इज इवॉल्व्ड फ्रॉम RNA, राइट. सो दोनों जेनेटिक मटेरियल हैं, बट द स्टेबल वन, द गुड वन विच इज प्रेजेंट इन good number, it is DNA. And for that purpose we have to know for the structure of DNA in detail, because ये पूरी प्रोसेस हम तभी समझ पाएंगे जबकि हमें डीएनए का स्ट्रक्चर बहुत अच्छे से पता होगा. आरएनए का स्ट्रक्चर भी हम देखेंगे. एंड एक चीज जो है वो ये है कि आरएनए जो है एक बैड जेनेटिक मटेरियल होने के साथ साथ उसके अदर रोल्स भी हैं. व्हाट इट इज अ मैसेंजर, इट इज एन adapter, it is a catalytic molecule, right? And it is having the structural role as well. So the point is that RNA, it is a genetic material in some viruses, and the viruses they are known as retroviruses. Besides the function of genetic material, it is having the another functions as well. It works as a messenger, it works as an adapter, a catalytic molecule, and it is having the structural function as well. Now come to the next point here, and that is all about the DNA. डीएनए की फुल फॉर्म हमें पता ही है, डीऑक्सी राइबो न्यूक्लिक एसिड, राइट. एंड इट इज अ लॉन्ग पॉलीमर. Need not to discuss what the polymer is, children. Monomer and monomer constitute the polymer, right? Monomer and monomer constitute the polymer. So it is a long polymer. और polymer किसका है ये? It is a polymer of nucleotides, right? अगर हम न्यूक्लियोटाइड्स का स्ट्रक्चर समझ लेंगे तो फिर हमें पता चल जाएगा कि what the structure of the DNA is, because it is nothing but the polymer of the nucleotide, right? Now, the length of the DNA, it is defined as the number of nucleotides which are present. And the pair of nucleotides means base pairs. The length of the DNA, it is determined by the number of nucleotides or base pairs. So यहाँ पर आप example दे सकते हो. Example in the form of bacteriophage. right? It is having 5386 nucleotides. Another bacteriophage, lambda, it is having 48502 nucleotides. Another example you can give here, that is what? Escherichia coli. It is having 4.6 into 10 raised to power 6 base pairs. And let us come to us children, human. And the haploid content is given, that is 3.3 into 10 raised to power 9 base pairs. So these many points you have to remember, right? For the objective questions. E. coli, it is having these many base pairs. Human, it is having the haploid content in terms of 3.3 into 10 raised to power 9. Bacteriophage 174, it is having 5386, and the lambda, bacteriophage lambda, it is having 48502 nucleotides. Now, again, DNA is nothing but a polynucleotide, a polymer of nucleotides. अब देखें, that we have discussed, right? It is made up of molecules called nucleotide. अब nucleotide जो है, इसके 3 components हैं. और 3 components में क्या क्या है? 1 nitrogen base, right? Second, phosphate group, and third, sugar. It can be ribose, can be deoxyribose. So ये 3 components मिलके एक न्यूक्लियोटाइड का फॉर्मेशन करते हैं. तो हम कह सकते हैं that a nucleotide has 3 components: a nitrogenous base, a pentose sugar, it can be the ribose sugar or deoxyribose sugar, and a phosphate group, right? Now, अब देखिए, the nitrogenous base, they are of 2 types. Purines and pyrimidines, right? Adenine or guanine, they are purines. And cytosine, uracil, and thymine, they are pyrimidines, right? So the nitrogenous bases, all in all, they are of 4 in number: adenine, guanine, cytosine, and thymine. In RNA, it is uracil in place of thymine, and they are grouped under purines and pyrimidines. Now, here you can see the nitrogenous, it is attached with the sugar. The nitrogenous base, it is attached with the sugar here, and here the phosphate group, it is also attached. When the nitrogenous base, it is linked with the sugar, right? Then the nucleoside, it is formed. And here the linkage is N-glycosidic linkage. So you can say here the nitrogenous base, it links with the sugar through this linkage and glycosidic linkage, and the nucleoside it is formed, right? And you can name here the nucleoside in terms of adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. You can find it in the diagrams and all. I'm not writing this just to save the time. And for now, just remember that the nucleoside they are formed by the linkage of nitrogenous base and the sugar, and the bond it is of glycosidic type, glycosidic linkage, because ये important है. अब देखिए, when the phosphate group, right, it is linked to the 5' OH of a nucleoside, then what is formed? A nucleotide is formed, and the bond is phosphoester bond. I am writing this with another color here, children. The bond is phosphoester bond. इसमें कोई कॉम्प्लेक्सिटी नहीं है, देखिए. यू नो न्यूक्लियोटाइड का स्ट्रक्चर हम देख रहे हैं, 3 कंपोनेंट से मिलकर बना होता है. वन इज नाइट्रोजनस बेस, सेकेंड इज शुगर, एंड थर्ड वन इज फॉस्फेट ग्रुप. पहले नाइट्रोजनस बेस शुगर के साथ ज्वाइन करता है और न्यूक्लियोसाइड बनता है. द लिंकेज इज एन ग्लाइकोसिडिक लिंकेज. और अब न्यूक्लियोसाइड में फॉस्फेट ग्रुप अटैच होता है तो वो न्यूक्लियोटाइड बन जाता है, राइट. एंड द बॉन्ड इज फॉस्फेस्टर बॉन्ड. ठीक. तो अभी हमने 2 बॉन्ड्स देख लिए. अब देखिए, न्यूक्लियोटाइड का स्ट्रक्चर हमने देख लिया, और हमने पहले देखा कि डीएनए इज अ पॉलीन्यूक्लियोटाइड, यानी न्यूक्लियोटाइड्स जो है अटैच रहते हैं एक दूसरे से. So the two nucleotides they attach through a bond that is called as phosphodiester bond. And the phosphodiester bond we have discussed, in the 11th standard in the chapter biomolecules, right? So here you can say that the two nucleotides they are linked through 3-5- phosphodiester bond, and the more nucleotides they can join in this manner, children, right? Now इस स्ट्रक्चर से हमें क्या पता चलता है? इस स्ट्रक्चर से हमें पता चलता है कि a polymer is formed in this way by the linking of the nucleotides through the phosphodiester bond, और उसके एक एंड पे फ्री फॉस्फेट मोटी होती है, एंड एक एंड पे a polymer it is thus formed has at one end a free phosphate moiety at 5' end of ribose sugar, and that is how it is referred to the 5' end of polynucleotide chain. And similarly at the other end of the polymer, the ribose has a free 3' OH group. So the end is known as 3' end. Let me clear here this structure if it is given in the diagram. Okay, I'll make you this diagram, a quick diagram for you. I just want to show you the 5' and the 3' and, children, right? So here you can make like this. यहाँ से nitrogenous base protruded है like this, right? You can make, और स्ट्रक्चर आप एक बार बना के देख लेंगे तो आपको और ज़्यादा क्लियर हो जाएगा. यू नो, बायो स्टूडेंट्स को केमिस्ट्री थोड़ी सी टफ लगती है, और मुझे भी लगती है. So this one and this one. You know, just see the structure here, children, right? A polymer thus formed has one end a free phosphate moiety at 5 dash end of ribose sugar, and that is why this is referred as the 5 dash end of polynucleotide. And at the other end of the polymer, the ribose has a free 3 dash OH group, and that is how it is referred to as 3 dash end of the polynucleotide. क्योंकि हम बताएंगे, जब हम स्ट्रक्चर देखेंगे तो हम पढ़ेंगे दो स्ट्रैंड्स होते हैं, एंटी पैरेलल होते हैं, राइट? वन इज 5 डैश 3 डैश एंड सेकंड वन इज 3 डैश 5 डैश. सो ये 5 डैश और 3 डैश का डिस्कशन ये है कि 5 डैश एंड पे फ्री फॉस्फेट ग्रुप होगा एंड 3 डैश एंड पे OH ग्रुप होगा, 3 डैश OH ग्रुप विच इज रेफर टू द 3 डैश एंड. And this is what? This is the structure of a strand, right? Like this is the polynucleotide chain, you can say. ये bases हैं, sugar, right? And the phosphate group. So that is how the polynucleotide chain it is formed. अब थोड़ा सा आगे का डिस्कशन लेते हैं यहाँ पे. यू नो, आरएनए में क्या होता है? आरएनए में दो चेंजेस होते हैं. What? The sugar type is different. Ribose sugar. Second, a free 3 dash OH group it is present, right? A free OH group it is present at 2 dash end, sorry. A free OH group it is present at 2 dash position of ribose. तो आरएनए में दो चेंजेस हो गए चिल्ड्रन. व्हाट? द शुगर टाइप इट इज ऑफ राइबोज, and the second one is a free OH group it is present at the 2 dash end of the ribose sugar. Now, you know, the DNA which we are discussing here, it is discovered as an acidic substance present in the nucleus. By whom? By Friedrich Miescher in 1869. And उन्होंने इसको नाम दिया न्यूक्लिन. This is what the beauty of this chapter, children. You know, डिटेल में हमें देखना पड़ेगा एक एक पॉइंट, बिकॉज जब हम ऑब्जेक्टिव करते हैं तो ऑब्जेक्टिव तो कहीं से भी आ सकता है. दैट इज वाई. So here DNA it is discovered as nuclein, which at that time he told that it was an acidic substance, right? Friedrich Miescher, 1869. After that, in 1953, James Watson and Francis Crick. Two scientists was there. They gave the structure of DNA. They proposed a very simple but famous double helical structure of DNA. Come here. So, what we have discussed till now, children, what we are discussing the molecular basis of inheritance. A quick review we can see here. DNA is nothing but the polynucleotide, a polymer of nucleotide, right? And the nucleotide, it is formed by three components, and the three components they join each other by different bonds that we have discussed here. Base, it joins sugar with glycosidic linkage, and the nucleoside is formed, and after that nucleoside it joins phosphate through phosphoester linkage, and the nucleotide is formed, and the two nucleotides they are joined by the phosphodiester bond. उसके बाद हमने देखा कि पॉलीन्यूक्लियाइड न्यूक्लियोटाइड की जो चेन बनी है उसके उसके एक एंड पर फ्री फॉस्फेट moiety होती है, and that is what the 5' end of ribose sugar, and the similarly at the other end of the polymer the ribose has a free 3' OH group which is referred to 3' end of the polynucleotide chain. और आरएनए में दो चेंजेस होते हैं, एक तो शुगर टाइप और एक फ्री OH group at the 2' position of the ribose sugar. Now come here. You know, 1869 में न्यूक्लिन बोला, एसिडिक सब्सटेंस बोला उसको डीएनए को. बाद में 53 में 1953 में वॉटसन एंड क्रिक ने based on the X-ray different diffraction data, you know, साइंस में जब भी कोई चीज प्रूव होती है तो एक साइंटिस्ट ही उसको प्रूव नहीं करता. मींस कहने का यह है कि जो भी थ्योरी बनती है इट इज बीइंग कंट्रीब्यूटेड बाय मैनी साइंटिस्ट, क्योंकि स्टेप बाय स्टेप उनको वो लिटरेचर पता चल गया तो वो फर्दर आगे बढ़े, फिर उनको पता चल गया तो वो फर्दर आगे बढ़े, इस तरह से होता है. तो दैट इज हाउ यू कैन से हियर दैट इन 1953 जेम्स वॉटसन एंड फ्रांसिस क्रिक बेस्ड ऑन द एक्स-रे डिफ्रेक्शन डेटा produced by Maurice Wilkins and Rosalind Franklin, right? They proposed a very beautiful structure, very like it is what? It is a famous and simple structure which is called as the double helix model of DNA. He proposed the double helical structure of the DNA. And one of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. Here you can see here. This is one polynucleotide chain, this is another polynucleotide chains. They are anti-parallel in fashion. One is 5' 3', the another one is 3' 5', right? And here we will discuss this in detail. Let us come here. You know, इसमें एक important चीज और है. This proposition was also based on the observation of Irwin Chargaff. उन्होंने क्या बताया? उन्होंने बताया कि डबल स्ट्रैंडेड डीएनए में the ratios between adenine and thymine and guanine and cytosine, they are constant and equal ones, right? So here the base pairing confers a very unique property to the polynucleotide chain. All in all, what we have discussed in this slide, that in 1953 children, James Watson and Francis Crick, based on the X-ray diffraction data of Maurice Wilkins and Rosalind Franklin, gave the double helical structure, which was also based on the observation of Irwin Chargaff, right? And what the observation of the Irwin Chargaff is that here the ratios between adenine and thymine and guanine and cytosine that are constant and equal ones. आप देखिए हम बार बार बोल रहे हैं कि the double helical structure they gave the simple and very good DNA structure, double helical structure. What actually this double helical structure is, and you know, this one, this is the double helical structure. What the salient features of the double helical structure of DNA is, it is made up of two polynucleotide chains. It is made up of two polynucleotide chains. Polynucleotide chains क्या होती हैं? That we have discussed, right? You know, polynucleotide, which is the polymer of nucleotide, that we have discussed how it is formed. और two polynucleotide chains मिलकर एक DNA बनाती हैं. It is made up of two polynucleotide chains, where the backbone it is made up of sugar and phosphate. Right? How the backbone it is made up of sugar and phosphate. Let us discuss here. This one. The backbone it is made up of sugar and phosphate. You can see here, right? Second, we are discussing the double helical structure, right? First point is that the DNA is made up of two polynucleotide chains, and here the backbone it is made up of sugar and phosphate. Second is the two chains they are anti-parallel in polarity, right? What it means? It means if the one chain has the polarity 5 dash 3 dash, then the other has 3 dash 5 dash, right? Now, what the 5 dash and 3 dash is that we have discussed. 5 dash पे एक phosphate moiety and 3 dash पे एक OH group. That is why they are called as this free groups are there. Now, the bases, cytosine, guanine, adenine, thymine, these are what? These are bases. The bases in two strands, they are paired through hydrogen bonds. These dotted structures, they are hydrogen bonds. So adenine base pairs with thymine, cytosine base pairs with guanine, and between cytosine and guanine, the three hydrogen bonds are there. And adenine, it joins the thymine by two hydrogen bonds. This is the structural point. साइटोसिन ग्वानिन के साथ थ्री हाइड्रोजन बॉन्ड्स से जुड़ता है एडेनिन थाइनिन के साथ दो हाइड्रोजन बॉन्ड्स से जुड़ता है राइट और इसी का रिजल्ट क्या हुआ प्यूरिन इट कम्स विद pyrimidine right the two chains they are coiled in right handed fashion the two chains they are coiled in right handed fashion. Let me draw a simple diagram of you for you. The two polynucleotides chains 5 dash 3 dash 3 dash 5 dash. right? That is how they are anti-parallel. The backbone, it is made up of sugar and phosphate. And the, here the adenine, it base pairs with thymine by two hydrogen bonds, right? And you can say here the cytosine, it base pairs with guanine by three hydrogen bonds. So this is the structure. Now the pitch of the helix, it is what? It is 3.4 nm, and you know 1 nanometer equals to 10 raised to power minus 9 m. So यहाँ से लेकर यहाँ तक that is 3.4 into 10 raised to power minus 9 m, और यहाँ से लेकर यहाँ तक 10 base pairs होते हैं. Adenine, it base pairs with thymine. Cytosine, it base pairs with guanine. Three hydrogen bonds you can make, two hydrogen bonds you can make, right? और अब यहाँ से लेकर यहाँ तक कितने होंगे? 10 base pairs. So दो base pairs के बीच में कितनी दूरी होगी? It is divided by 10, right? So लगभग 0.34 nm or 0.34 into 10 raised to power minus 9 m. So that is how you can make the length, you can detect the length of the DNA, right? How? Because we know that the length between two base pair, it is 0.34 into 10 raised to power minus 9 m or 0.34 nm. The pitch of the helix, right? The pitch of the helix, it is 3.4 nm, and here roughly 10 base pairs in each turn. So the distance between two base pair in a helix, it is 0.34 nm. Or you know the base pairs, they stacks over the other, they stacked one upon another like this, and the presence of the hydrogen bonds, right? 2 3 2 3, right? ये जो है स्ट्रक्चर डीएनए को स्टेबिलिटी देता है. What the points? अगर आपसे कोई पूछे कि what the points for the stability is, as the base pairs they stack over the other in double helix, or the presence of hydrogen bonds, right? They give additional stability to the helical structure. तो ये डीएनए का स्ट्रक्चर है जो कि डबल हेलिकल स्ट्रक्चर कहलाता है, एंड इट इज proposed by whom? By Watson and Crick in 1953. And the proposition of a double helical structure for DNA and its simplicity, right? It is very revolutionary. And उसके बहुत जल्दी ही, इसके तुरंत बाद जो है सेंट्रल डॉगमा प्रपोज कर दिया फ्रांसिस क्रिक ने. What the central dogma is that the information, it flows like this: DNA, RNA, and protein. This is what the central dogma is: DNA, RNA, and protein. The formation of DNA, the formation of RNA from DNA, it is transcription, and the formation of protein through RNA, it is translation. Now, I just want to show you one more diagram, you know, and that I just want to make for you. जिससे आपको ये स्ट्रक्चर और ज़्यादा क्लियर हो जाएगा। और ये जो डायग्राम है अगर आप खुद बना के देखेंगे तो बहुत अच्छी बात होगी। Double helical structure of DNA we have seen here, right? And the two chains, they are anti-parallel in fashion. What the anti-parallel is that we have discussed now. एक polynucleotide chain तो मैंने आपको बना के दिखाई थी. The same chain it is, right? And here you can make like this. ये डायग्राम बनाने से स्ट्रक्चर आपको और ज़्यादा क्लियर हो जाएगा. The DNA structure, which is the base of this chapter, children. आरएनए में क्या चेंजेस हैं? आरएनए में शुगर का चेंज है एंड एक एडिशनल OH ग्रुप प्रेजेंट है 2' एंड पे, ये चेंज है. तो ये भी आपको याद रखना है बिकॉज जब हम पढ़ेंगे कि वाई आरएनए इज अ बैड जेनेटिक मटेरियल एंड डीएनए इज अ गुड जेनेटिक मटेरियल, तो वो आपको बताना पड़ेगा. दैट इज हाउ, लाइक ये थोड़ा सा ऐसे है. एडेनिन इट, आप ऐसे बना सकते हो उसको. Adenine, it base pairs with thymine, right? Double bond. And here thymine is base pairs with adenine. Here guanine base pairs with cytosine 3. And here the cytosine, it base pairs with guanine. And here what? OH group. Because the two chains, they are anti-parallel like this. And NEET में नो जो डायग्रामेटिक क्वेश्चंस भी आ जाते हैं. दैट इज़ हाउ डायग्राम्स जो हैं वो इम्पॉर्टेंट होते हैं, आपको पता ही है. See, this is the diagram which I have made for you, right? एंड हो सकता है इन लाइक इंक्लिनेशन में थोड़ी सी प्रॉब्लम हो गई हो, बट स्टिल this is one polynucleotide chain, this is another polynucleotide chain, right? What we have to understand here, जब हम बोलते हैं the two strands they are anti-parallel in fashion. So here you can see the 5' end, 3' end, the other one is 3' end and 5' end, right? And here the base pairing you can see between the two base pairs, like the adenine it base pairs with thymine and the guanine it base pairs with cytosine. The number of hydrogen bonds you should have to remember. And the second thing is what? Here the pitch we have to discuss, like the distance, the pitch of the helix is 3.4 nm, and the distance between two base pair it is 0.34 nm. That is how. So here we have discussed the structure of the DNA. The diagram it is also there, right? The same diagram which I have meant for you. The helical structure you can see, the right-handed fashion you can see here, the you know the two polynucleotide chains where the backbone it is constituted by sugar phosphate and the bases they are project inside, and the two chains they are coiled in a right-handed fashion. So you can see here DNA ladder, double helix, right? Children, these are the bases which form the nucleoside, and after that the nucleoside it is attached with the phosphate group to form the nucleotide, and the polynucleotide the two polynucleotide chains, they form the DNA, right? So this one, and I think the structure should be clear to you. अगर मैं आपको एक क्विक रिव्यू दूं, एक्चुअली मैं चाहती हूँ कि आपके सारे पॉइंट्स क्लियर हो जाएं। तो अगर हम डबल हेलिकल स्ट्रक्चर की बात करेंगे तो फिर क्या होगा? टू चेन्स विच आर एंटी पैरेलल इन फैशन, coiled in a right-handed manner, एक हेलिक्स की डिस्टेंस दैट इज 3.4 nm, एंड यहाँ से लेकर यहाँ तक 10 बेस पेयर्स आप बना सकते हो। तो द टू बिटवीन द टू बेस पेयर्स द डिस्टेंस इज व्हाट? 0.34 nm. 1 nm equals to 10 raised to power minus 9 m, right? Adenine base pairs with thymine, cytosine base pairs with guanine, and the hydrogen bonds they are present in between. The three hydrogen bonds between cytosine and guanine and adenine and thymine के बीच में two hydrogen bonds, and the two polynucleotide chain, they are anti-parallel. So these many points you have to remember for the structure of the DNA, the presence of the hydrogen bonds, and the presence of base pairs in such a manner that one stacks over another like this, one stacks over another like this, they give the stability to the structure of DNA. आरएनए में जो चेंजेस होते हैं एक बार फिर से देख लीजिए। आरएनए में क्या चेंज होता है? प्रेजेंस ऑफ एडिशनल OH ग्रुप at the 2' end position, sugar is ribose, एंड आरएनए में थाइमीन की जगह यूरेसिल भी होता है। द थाइमीन इज इट इज रिप्लेस्ड बाय द यूरेसिल, यू नो। यूरेसिल इज व्हाट? इट इज अ pyrimidine. सो नेक्स्ट वीडियो में हम क्या पढ़ेंगे? हम पढ़ेंगे डीएनए पैकेजिंग एंड द एक्सपेरिमेंट्स फॉर द सर्च फॉर genetic material which is the DNA, the experiments which actually proved yes DNA it is the genetic material. and अगर आपको ये वीडियो अच्छा लगा तो आप इसको लाइक करिएगा, शेयर करिएगा. Keep watching the videos and have a very good day.